Okay, and we are back talking about seeds. Okay, seed storage is really important, especially in a commercial greenhouse or anytime you're trying to grow lots of seeds. You want to maintain the proper temperature, you want to maintain the proper humidity, you want to make sure that the embryo remains alive. So you don't want it to be too cold, you don't want to be too warm. You certainly, I think the biggest thing in seeds is you don't want to be too moist. Otherwise, in the bag, your seeds will begin to germinate, and as you can imagine, that is not what you want. You don't want a bag of partially germinated seed. Again, some things people will do before they plant, especially in a commercial greenhouse, is they will do a cut test to see if the embryo is present. They will do a float test. Bad seeds will float. Some very high dollar greenhouses will even x-ray to make sure the embryo is present. They will do a germination test. This is probably what everyone does. Essentially, you're going to take 10 seeds, do that 10 times so that you have 100. You're going to put them all down. You're going to wrap them in a wet paper towel looking thing, but you can just do it under a wet paper towel. You're going to put them in the refrigerator. Every 24 hours, you're going to see how many of them germinate for about 72 hours, and that's going to give you a percentage of germination. That's also going to tell you how many to plant. If you have old seeds, let's say you're getting 75% germination, then you can determine if you need new seeds, and it'll help you determine how many seeds to plant per pot. There's also some tetrazolamine tests, some things that turn red, some embryo tests. Again, place in a moist paper towel, check for signs of activity. This is pretty common. Seed dormancy. There's different ways to break dormancy. Mostly just moisture breaks it, but some seeds have to be scarified, it's called, which means you have to rub them on something. You have to physically break the seed coat. Cucumber, of all things, is incredibly hard to get to germinate, which is crazy because it's a weed and it's everywhere. So there are certain seeds that have to have certain things done to them. Like I said, the woman with the elephant, it has to be ingested by an elephant and pooped out. So a lot of times breaking dormancy isn't as easy as you think, but most of the time in a commercial greenhouse, you already know how to do that, and it's usually pretty self-explanatory. Seed coat dormancy prevents water or atmospheric gases from reaching the embryo and initiating germination. Sometimes that can be broken by scarification, which is rupturing the seed coat. It's usually like sandpaper. You will usually put the seeds on sandpaper and scratch them. Stratification. Also, you have to sometimes expose the seeds to a chilling temperature. So you have to put them in the paper towel and then put them in the refrigerator, so they have to be cold for a while. And sometimes you have to do both, scarification and stratification, to get things to break their dormancy and germinate. As you know from being in the greenhouse, it's not as easy to just plant a seed. They don't always germinate the way you think. Germination steps, they're going to imbibe water. They're going to take in water. That's going to activate some of the hormones, which will break down the food, increase respiration, and help the food materials move around. So essentially, the embryo starts to grow. You get a differentiation between your shoots and your roots. Your seed coat ruptures. The photosynthetic tissue starts emerging against gravity. Usually, sometimes it grows towards different wavelengths of light, and sometimes it grows away from gravity. So that's phototropism versus just another kind of tropism that we'll talk about. And it's either growing away from gravity or it's growing towards light. And usually the radicle extends into the soil. Your root grows away from the light and towards gravity. So here's some seeds growing. These guys are trying to get their little green tissue above and their root tissue below. That's a wheat seedling. Here they are taking in water. Their roots are going to grow. This is for a dicotyledonous. Their roots are going to grow, and then you're going to get it push up here, the roots and the dicots. These are the dicots, remember? Seed germination requires pretty specific environmental requirements. You need to know that if you're growing something from seed. How much moisture does it need? What's the temperature it needs? What kind of aeration does it need? What kind of soil does it need? This is usually found on the back of a packet if you get commercial seeds from like Ball Seed or Park Seed or just at Walmart or something. That we usually tell you that. They're not kidding. You should really read that. It requires specific light. Some need to be planted really shallow because they grow towards the light. So there needs to be enough wavelength so they can grow towards the light. Some require complete darkness, so you have to stick it way down in there, right? You have to get the seed like 1.5 inches. So again, cannot recommend reading the back of the packet enough. So are there pathogenic organisms? So sometimes you can overwater your seed and you will get damping off. It will start to get moldy. So you want to make sure that you don't overwater them and keep them in a moldy environment. Sometimes there's a toxic amount of salt in your water. So you don't want to use a ton of fertilizer when you first germinate them because you could over-germinate. I mean, you could over-fertilize them. Vegetative propagation. Okay, so this is essentially what we talked about in the lab and what you guys did. This maintains the genetic stock of the mother plant. We call it a mother plant. It's not necessarily female. We just call it that. Cultivars maintain asexual propagation. Cultivars are called clones. So essentially we are cloning the plants. All plants of the same clone are genetically identical, and they are an extension of the original stock plant. So you have vineyards that have 200-300-year-old stock plants. It's very interesting. Apple orchards, their stock plants are hundreds of years old, what the stalk is on, and then they graft new varieties on, but they never dig up the roots of the old one. Vegetative propagation, you also get chimera mutations, which a lot of people try and do. Look how cool that leaf is. Essentially, it's variegated varieties, thornlessness, peach fuzziness. It's also sometimes a way of reducing seed size, so you can get things that are seedless. Chimera mutations are determined by the location of the shoot apex. Where the mutation occurs determines the type, if it's stable, if it's not stable, it's going to be here. So this mutated cell on the mericlinial is not going to be very stable. This periclinial is going to be a little bit more stable because it's right here in the center. So these are just weird. Mericlinial, periclinial, and sectorial are just weird. The mutation occurs in the growing part of the apical meristem. Apomixet, basically this is seed formation and germination. Let me move this over here. So sometimes you will not get the proper meiosis happening and you will get a mutation. Embryo has same genetics as parent plant, often develops from citrus tissue. Seedlings go through juvenile and mature transitions as a normal seedling would. This is a lot of the things they do in citrus. Clones. Disease problems. Systematic disease can be passed from generation to generation through propagative material. Entire clone can eventually become infected and you have to start over and genetically breed back the clone that you wanted. Typically, this can be overcome with a heat treatment that you use with your clones. A shoot tip culture, some enzyme, some ELISA, looking at a quick amino assay. All of this you would do if you were a huge production greenhouse. You want to do this all the time to make sure that your clones are not developing anything as you're propagating them. Okay, propagation by cuttings. You guys did this. Propagation by cuttings. It's probably the most typical thing that happens in production greenhouse management. You're going to take a piece of vegetative tissue. Under the right environmental conditions, it's going to regenerate the missing parts, the roots, the leaves, whatever. It's typically a stem situation. You can use leaves, you can use leaf buds, you can use roots, but by and large, we are propagating stems of things. Okay, stem cuttings. This is exactly what you did in class. Essentially, the most common is to just take a branch like this, the top branches of the plant. The long stem can be cut into more than one piece, especially if you go with an auxiliary bud. Excess and lower leaves should be removed at this point, and do not remove too many stems of the mother plant because it will kill it because it won't be able to photosynthesize. Most are propagated in a greenhouse using a porous root medium because, again, you're very concerned about disease at this point. High humidity. So this is a commercial greenhouse, and these are cuttings, and this is definitely what it looks like. You will go in, and it will just be like mist will hit you. It's crazy. It's reduced light. It's a heated, these pads are heated, and it is way more of an art than a science. People know exactly what humidity this should be at. Sometimes they're set with computers and you will just get this high misting and then it will shut it off and hopefully